This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 106, recorded November 2nd, 2010. Hi everybody, I'm Vincent Yellow, and this is TWiV, and that is Dickson de Palmier.
Indeed.
Right across the desk from us.
Yellow.
We're doing another Virology 101 today.
Good.
We have the video running. There's the camera over there. And on our screen is Rich Condit from Florida.
Hi, fellas.
How you doing?
I'm doing great.
What is the hi fellas from? Is that from a TV show?
I don't know.
No, it's just
greeting.
It's greeting from the south.
It just came right out.
I'll bet you it's warmer where you are right now than it is where we are.
Let me see. My weather bug says it's 73.
Oh
and
my
it's
God!
Look at him. He's got
a
a light shirt on.
It's well below 50 degrees here.
And out my window, they just killed a palm tree. Actually, it died. And they're burying it. And that's like the fourth palm tree they've tried to put in that spot. And if I were a palm tree and they tried to put me in there, I'd be shaking in my boots.
I'd call in the EPA.
Yeah, there's something going on in that.
There's got to be.
Here this morning it was 1 degree centigrade, at least where I was.
Right.
Same with you. It's just above freezing.
That's right.
If you go a little bit north where Professor Silverstein lives, it's freezing.
It is freezing.
What do you think about that, Rich?
I think that's just not good at all.
He lived in Buffalo. He lived in Buffalo for 12 years.
12 years.
Rich, you know what's wrong with this scenario is that all of us are now heading down to you as we speak. There's a caravan of snowbirds that are flying south as we speak.
Well, I'm heading to Paris today.
You are? Well, that's a nice
where it's
venue.
It's 60 degrees, I think.
That's a cool venue.
But I'm wearing a ski jacket. I'm going to look silly down there. Right?
Tomorrow I'll be in Washington, D.C., so that's maybe
a little bit warmer. All right, enough of the weather.
Right.
Our listeners don't want to hear the weather. What they want to hear about is virology.
Indeed.
And let's continue with part 2 of making viral DNA.
So this is Virology 101.
Virology 101.
Okay.
Basic stuff where you ask the questions.
I do.
We're the professors, Rich and I.
Okay.
You can ask whatever you want. You get a free education.
This is great.
We try and answer.
Something I've always needed, of course.
Yeah. So what I'm going to do now is let Rich see my screen, share the full screen, and then we're going to go to...
So since this is about viruses,
it is
the whole
about
thing
viruses.
about viruses, and we've already talked about the number of different ways that they can replicate, 7 in all. I keep referring to that
right.
for fear I will
I think you have it now. I think you have it.
So now we have double-stranded DNA viruses. This sounds a little bit like a host cell.
Yeah, very much.
Okay, so which viruses fall into the double-stranded DNA category?
Well,
Vince?
I have a picture coming up in a couple of slides. Can you wait?
I can wait.
Vincent,
give us a familiar...
Vincent, I can't see your screen.
You can't see my screen?
Nope.
All right, let me go back and retry it. Sometimes this Skype share full screen. Okay.
Aha, now something's going on.
There you go.
You see it now?
Oh, yeah.
Okay.
You see a nice single-stranded DNA there?
I see a double-stranded.
I see a double-stranded.
You can't fool me.
Or me.
That's double-stranded DNA.
I may not know much about virology, but I know a double-stranded.
All right,
I see it.
I wanted to make sure Rich was seeing it, because he could have said, yeah, I see it, and not see it,
Right.
And you...
Oh, I see it.
But your parasites have this double-stranded DNA stuff.
Hey,
that's the molecule of our episode. We showed this last time, a nice double helix.
Yep.
2 strands. And let's take another look at what those strands are made of. These are these base pairs. 4 different bases, adenine, thymine, guanine, and cytosine, which pair in specific way. That's the informational part of this molecule. Indeed. It's got a backbone of phosphate and deoxyribose. Phosphate shown. Well, I can't use my... Oh, look, I can use my pointer.
Absolutely.
The phosphate's here. Here's the ribose, a 5-carbon sugar. Makes a nice backbone. So there are 2 backbones because it's double-stranded, and then the base is in between. And last time Rich told us all about these bases. So you should go back to, what was that, TWiV 96, I think?
Oh, I don't know.
I think that's right, TWiV 96.
Yeah, it was just short of TWiV 100.
So that's what the molecule looks like. And if you pull it apart... If you denature it or melt it, those are the different ways we can say that.
Yes.
You can make single strands, which can we can then copy each single strand to make a new double strand molecule. So in this picture, We've taken our double-stranded DNA. It's being pulled apart here by unseen forces.
Indeed.
Which could be enzymes.
Enzymes.
Usually it's an enzyme. Do you know what the name of that
Helicases.
is? Ah.
I'm going to ask Dickson. I'm sorry.
No, that's okay. I would have.
That's all right. I'm happy to have answers from Florida.
To reveal my ignorance at this level is not an oversight on my part. I can do it.
No. Ad libitum. Right. So helicases melt it in the process of replication. Now you have two single strands, and another enzyme comes in, the polymerase, and makes a second copy. So that's DNA replication, which we talked about last time. And the fact that only certain bases go together, G with C and T with A, means that you can easily make another strand.
You can.
And, of course, Watson and Crick mentioned that.
They did mention it.
Do you know what year that was, Dickson?
1958, I think.
No.
Wasn't it?
It was the year of my birth.
1950.
1901, you're going to say.
If I knew how old you were, I would have guessed it. I thought it was 58, but it was
53.
53, even earlier than that then.
Yeah. Okay. DNA replication, to summarize, we're going to just review. Viruses must replicate their genomes, obviously. Otherwise, you can't make new progeny. Right? You seeing this, Rich? Everything good?
I'm seeing it. Everything's cool.
This always requires expression of at least one viral protein, sometimes many. So the simple viruses with small genomes can't encode a lot, so they only make one protein. Usually not a polymerase.
Seems like a very uninteresting virus at that
But
point.
the big ones encode a lot, like the ones that Rich works on. DNA is always made in a 5 to 3' direction.
Indeed.
With respect to the strand that is being synthesized.
That's right. The strand that's copied is copied in what direction?
From 5 to 3.
No, copied. It's synthesized. The new strand is made in a 5 to 3. But what is the template copied in what direction? You don't get it? How about you, Rich?
I'm guessing 3 to 5.
He's guessing.
Yeah.
He's not guessing. He knows the answer. That's not fair. You guys are setting me up for...
I'm not setting you up. It's okay. Yeah, the new DNA is made 5 to 3', and the template is copied 3 to 5'. It's a little confusing even for some of us old folk who've been around a while. And, of course, this 5 to 3' is a polarity imposed by the nature of the DNA, which we talked about last time.
Okay.
Finally, replication begins at an origin of replication, using a primer.
Right, which is sequence-specific, I understand.
It is. And then the host provides proteins that the virus doesn't encode. Right. And this was Rich's favorite slide, the 5' end problem. Want to review this for us briefly, Rich?
Okay, very quickly. So at the very top, you're showing a template strand in blue and RNA primers that are laid down in green because DNA polymerases can't initiate replication de novo, that is by themselves. They have to add on to a pre-existing structure, which is ordinarily supplied as RNA primers. So in the second part there, you see the same blue strand, and you see that the RNA primers have been now elongated with red DNA. And then the next step ordinarily is to excise the primers and then use the new 3' ends from the DNA, there's a bunch of them there, to elongate again to the point where now you have just a nick left between the DNAs that come up against each other, and it gets ligated. There's a problem, however, because you excise the primer from the extreme 5' end, you have a gap. Right. And if you try and do the same thing over and over and over again, you can never completely fill that gap, and that is an issue. As we described last time, somebody in the ultimate design of this goofed and left us with a problem. And so we got to deal with this problem, and viruses have clever ways of doing it. And a lot of the interest for me in, how viruses do this is in solving that problem. We're going to see several ways. One we already saw last time, just to review, and we're going to see some more again, is don't have any ends. Have a circular genome.
I think we have that.
Exactly.
Yeah. That's right. So we did talk about some solutions last time, one of them being a circular genome, one of them the parvoviruses having interesting structures at their ends.
Right.
And also two basic modes of replication that we see, a replication fork for these viruses, papillomas, polyomas, herpes, and retroviral proviruses. This is the way the cell replicates its DNA. And then strand displacement, adenoparvo and pox viruses. talked about this last time in detail, so we won't go in it because today we want to do new stuff.
Right.
The DNA polymerase, small DNA viruses don't encode an entire replication system, so they don't encode their own polymerase. They encode instead proteins that orchestrate the host. Don't you like that? Orchestrate.
Right, good.
The conductor.
Good.
Papillomas, polyomaviridae, and parvoviridae. The bigger guys encode most of their own replication systems, including the polymerase herpes, adenoviridae, and poxviridae. Last time we talked about parvos and polyomaviruses. Today we'll talk about herpes, adeno, and poxviridae.
So all of this talk between the virus and the host cell begs the same question as to the origins of viruses, then, doesn't it? Because you get so many varieties of ways of doing the same thing. The whole thing is that the cell is a black box. In comes the virus, out comes the virus,
Sure.
and it uses the host cell. But there are so many different ways of achieving this that it seems to me that there were multiple origins of viruses throughout evolutionary history. Isn't that a fair assessment for this?
Well, the cell does it one way, right?
One way. One way.
So if the cell was there first, you can imagine viruses evolving from it that used that way, but then went off and did different things, right?
As mutation rates
Assuming
occur.
that that's the direction that things happened in. Right.
You know,
Maybe not
Interesting
always.
question, though, actually, as
Very
to whether
interesting.
there have been a number of different independent evolutions of viruses. I'll bet you there have. Okay,
based
but that's
on all
just
of
a
this,
bet.
you know, I'm an innocent here. Come on, I'm
Yeah, sure.
not an innocent bystander necessarily, but I'm here to ask certain basic questions, and it makes you wonder just how all this came about, because it just,
given the variety of
It's so
different
elegant.
ways of doing it,
Yeah,
it's hard
absolutely.
to imagine a common ancestor.
and there must be tons of trial and errors that didn't work at all.
Yeah, that's a good point. So you're saying, for example, that there might have been single-stranded DNA genomes in organisms at some point? Yeah, I don't see why not. Unfortunately, we don't have evidence for it at
Or
this time.
other ways of repairing the nick, or living with the nick. Forget about repairing it. Let's just go on with our lives here.
Yeah,
What would happen under those circumstances, Rich? I mean, with a nick, you can't replicate again, right?
Right.
At least under the circumstances that
point.
we now know.
That's a good point.
So, you know, a lot of false starts to give rise to all of this stuff. So there must be some selective pressures to maintain all of these.
Well, you know, the viruses do it many different ways, and the cells now do it one way. So for some reason, cells are content with that way.
Well,
And I don't know
that's
what.
probably true, but they RNA edit, and they do lots of other things that have to do
Yeah.
with the messages after it comes out of the DNA. So do viruses RNA edit?
Yep, they do.
Ha-ha!
I think they did it before cells did. I'm just kidding. I have no way of knowing that. They do RNA edit.
Of course, the other perspective, Dickson, is that, in fact, the viruses came first, that there was a primordial soup
Sure.
that was like a giant soupy cell, if you like, and the
Yeah.
viruses were there first, and then we made cells out of that. Who knows?
Yeah.
Not
Well,
me.
Lynn Margulis has the opinion that heterotrophs arose first, that ate other things, and that
Define
accumulated.
heterotrophs for me.
Something like not a plant. You know, plants are autotrophs. They can
Okay.
sort of take
Okay.
care of themselves.
Right,
but
right, right.
heterotrophs are the other group that, like animals, you know, that need to eat something in order to survive. But I understand your point. But Rich, think about the early Earth and how hot things must have been, and how, you know, the extremophiles that we now know as, you know, the archaebacteria. All of that stuff had to take place in a milieu that we were totally unfamiliar with.
Yep.
And can't imagine what it would actually be like.
Right.
And yet, here it is.
Here we are.
Here we are indeed. So I can understand why people think that comets and all those other things brought, you know, pre-life molecules to the planet from these primordial clouds of crap that just accumulated, right?
Here we are actually evolved enough so that we can actually sit here and study ourselves and have this conversation.
Well, there you
Isn't that
go.
wonderful?
Well, yes, it is exactly. But, you know, one of the big findings that we had by sending out a rocket to capture the comet dust and bring it back to find out how old comet dust is, to see whether or not they could have played a role in something like this, proved that they were 1 million years older than the formation of our solar system. So 1 million years down the road,
It's not a lot.
it's not a lot. Of course it isn't. But it didn't take part in the comets formed after the solar system began to condense up into planets. And that's an interesting part. I mean, this takes you right back to the very beginning.
Yeah, dust always comes second.
You
move in, then the dust comes.
All we are is dust in the wind. I love that song anyway. So
here's the topic for today, double-stranded genomes. We actually talked last time about single-stranded DNA genomes.
Right.
And today, double-stranded DNA. Actually, the polyomas, the circular double-stranded genomes, we talked about last time. Today, we're going to talk about linear genomes. And again, the information flow, you have a double-stranded DNA. mRNA is made from it, which is translated into protein.
Right.
The DNA then has to replicate and make more of itself, which is what we're looking at here. And I think first we're going to look at the adenoviruses. They have double-stranded linear DNA genomes shown here, 36 to 48 kilobase pairs long. And they have a protein at the 5' end of each strand. It's called terminal protein, TP. It's covalently attached to the DNA. Now let's see why that's there. That's there because it's used as a primer.
Why? Why?
Yes, it is a why. It's a why.
Because if you don't do that, you're stuck with the problem.
Well, there you go.
Right?
You've got this little bit that you haven't filled in. Here's the way to fill it in.
You don't
need to fill it in.
Exactly. You use a protein, you solve the problem. So why haven't all viruses done this?
Why haven't we done this?
It works for me. Maybe it doesn't work for you.
That's right.
You've got another way of doing it.
There
That's
must be a negative evolutionary reason why we don't do this, right?
Well, maybe it restricts us in what we can do then.
Let's explain it. So here at the top we have a double-stranded DNA. Here's the priming step. The polymerase is attached to this terminal protein.
Which has special characteristics, I presume.
It does. In fact, I'm going to show you the structure of it in a moment.
Whoa.
These are covalently attached initially, and then a protease cleaves them so they can be separated, but they remain associated. And then a C is linked to the terminal protein. My pointer goes away now and then. A C, the first base, is linked to the serine residue in the terminal protein. That serves as a primer. As the first base at the 3' end of the viral genome is a G. You can see that there. The C lines up with it, and that's the priming step. So there's one base primer, basically, and the protein makes it so that it can sit there properly, because one base would not normally be enough to be a primer by itself. So the polymerase then says, I have a primer here, begins to copy the genome. It displaces the other strand, which you can see bending away here, and the polymerase begins to copy the template, which is in the lighter blue in the bottom, and makes a new strand, which is shown in red. And that just goes down and copies the strand. Now, as it does, the displaced strand gets bound by a second protein called DNA binding protein, DBP. This is a single-strand binding protein whose function is to keep that displaced template displaced.
Right.
It's actually not a template. The template's the bottom. It's just the other strand which isn't being copied, right?
The negative strand.
Or is that the new one? Why is it shown here?
Better re-explain that one.
I'm a little confused here.
Wait a minute.
Help us out, Rich.
Let me get you back. I was cruising through something else. What's the problem?
We've got a yellow strand being displaced by the polymerase, and we're both confused as to which strand is actually the strand that we're concerned about.
So I think the newly synthesized one is the red. Okay?
Yes.
And the one that gets wrapped up in DBP is the displaced strand.
That's correct.
Right?
And then because of the inverted terminal repetition on this DNA, the two ends are complementary to each other, and you can form this panhandle structure.
Right.
Okay? And that is essential for priming the second strand synthesis, because now we got a terminal protein. That ends, the very end of that structure where the terminal protein is, looks for all the world identical to the end of the normal double-stranded molecule. So you just start priming synthesis again, and then in the next bit after step 5, you can see that unwound, and you just fill in copying that displaced strand, displacing the single-stranded binding protein, and you get another copy of the genome.
So that's the displaced strand. So when you did the first synthesis step, the red, you got a double-stranded product, one new strand with the template that's shown here. then the other displaced strand is copied separately.
So here's a stupid question, maybe not. You've got two covalently linked proteins that guide DNA synthesis for this double-stranded adenovirus.
Right.
When does that protein get made and incorporated into the next round of replication?
Well, it has to be made before DNA synthesis can occur, right?
So what programs that protein to be made?
Well, the viral genome, it's encoded in the viral genome.
So as all of this is going on, the protein is being synthesized and then it finds itself to the end and then sticks there?
Sure. Well, just imagine the incoming DNA of the virus comes into the cell, it goes to the nucleus.
Yeah.
mRNAs are produced, and some of those encode this Pol PTP precursor.
Okay.
That's all from the first one, because you can't duplicate that until you make this protein. So initially you have one viral genome, and that makes a bunch of Pol and terminal protein, and then those are in the cytoplasm. They're made in the cytoplasm.
Yeah.
Then they have to go back into the nucleus in order to prime viral DNA replication.
So if you had a drug that could interfere with just that protein synthesis, you could solve this.
Terminal protein would be a great adenovirus antiviral target.
Because this is specific for adenovirus.
Yeah.
We don't do this. Our cells don't do
exactly.
it. Yeah, sure.
Yeah, good point.
Do we have any drugs that do that?
No, but
Nope.
I don't think anyone's looked.
Got it.
It's a hard thing to screen. It doesn't have an enzymatic activity, right?
Yeah, but it has to be bound there by some process. It's not a spontaneous linkage, is it?
It's very It comes in with the polymerase, and then it's cleaved. And then when the polymerase starts moving, you can see here as the polymerase starts polymerizing, it leaves the terminal protein behind because it's been cleaved by the protease. It's a viral protease previously.
Sure. Oh, okay.
So the strands are made in two slightly different ways, you can see by this mechanism here. Now let's take a look at this interesting primer protein. Now, so this is a structure, a model of how this happens, which is learned from a bacteriophage, Phe29, which also uses
So
a primer.
Phe29 does the same thing?
It does. It has a protein linked at the end. It uses it as a primer, and the structure of that complex has been solved. And this is a model for how it happens. It's a little complicated, but I thought you might like to see this. On the left, we have a polymerase bound to the terminal protein. And here we have the first base, the C primer, sitting here. And this is believed to sit in the active site of the polymerase.
When you say it's been solved, you mean there's a three-dimensional
crystallography
of this. Okay.
Of course, we don't have a structure for every step.
No.
We have a structure for the first part on the left, the initiation complex.
Okay.
And then the rest is presumed to occur. This pre-terminal protein is thought to be flexible. It has a hinge because it has to bend back to allow the polymerase to start adding nucleotides. Now here in the second image, the blue is the template strand, and the red is the newly synthesized stuff. So the polymerase is now making a copy of the blue strand. Right. And as it does so, the pre-terminal protein hinges back to get out of the way, and then it eventually dissociates so that the polymerase can go down and make more. So it has to be intimately associated with the polymerase to begin with. It has to have that first C right in the active site, but then it has to move back to give room for the newly synthesized strand to move out, and that's what's shown here. So that's learned from Phe29, but we assume similar things happen with adenovirus. So maybe if you could target that hinging, if you could lock a drug targeting TP that locked it in this formation, that could be a good antiviral too.
But ultimately what you gather here is that there are certain themes throughout nature that are used for different things, and there are lots of hinge proteins that function in other ways as well.
Sure, absolutely.
And it's an elegant solution to a very seemingly difficult problem, but it looks like a simple solution, doesn't it?
It does.
Well, back to your question, Dickson, I wonder whether I would imagine that Phe29 and adenovirus have a common ancestor. It's a little hard to imagine this evolving twice, though maybe it did.
Right.
Who knows?
I agree. I think it's probably rare, but it could happen. But I think the idea of a common precursor makes more sense. And I'm sure people have looked at the sequences and tried to sort that out.
Right.
Yeah, nature never throws away things that work.
They only throw away things that don't work.
Well, they find another use for them.
Nature doesn't even throw things away that don't work.
No, you're right. You're right. The thing that astounded me most about that was the lens protein of mammals is a dysfunctional serine protease that's been crystallized.
So that's nature recycles as well.
It doesn't throw it. You're absolutely right.
In terms of mechanisms, yeah. But species go extinct because they can't survive anymore.
Well, that's a
but the
different
mechanisms live on in other animals.
Correct.
Right? One more look at this adenovirus. This is the DNA binding protein. This is the other protein that coats the displaced template. Remember?
That's a ribbon structure.
So on the left is the ribbon structure, and you can see it has a compact domain with lots of alpha helices, and that binds to the DNA. And it has a very extended C-terminus.
Are these basic amino acids?
Yeah.
Okay.
You would need that to bind to the negatively charged DNA.
Sure.
And the
helix proteins work in
exactly
the same way.
And on the right is how this protein binds to the displaced DNA. So we have a polymerase copying the blue template. A red strand is the new DNA.
I love this. This is great.
And then the displaced other strand is coated with this DNA binding protein, and that it sits on the DNA, and they snuggle into one another via that C-terminus. You can see there's a groove on the front part of the DNA binding protein, and that C-terminus fits into it. It's made to do this.
It's marvelous.
It's almost like a viral capsid protein.
It's like watching a car being assembled.
That's beautiful. I wish we could watch this, right?
You can, but if you describe it this way to people and they close their eyes, and you describe these shapes to them, it's as though you're reading it from a movie that you're watching.
What's amazing, though, is this is happening in a really crowded cell.
Yeah.
Really quickly.
And really quickly.
And it's amazing. It's all jammed together. It's not like open like in a picture like this.
I understand.
So the replication, to summarize, of adenovirus DNA, it's an example of strand displacement synthesis. It uses a protein primer. The origin is at both ends. I don't think we mentioned this yet. The end is the origin. Right. There's no specific sequence. It's just the end. Okay?
Like Montauk Point.
The DNA polymerase is viral, just like TWiV.
Yep. That's right. Keep going.
Other viral proteins involved include the terminal protein, which we looked at, the primer, and the single-stranded DNA binding protein.
Both of which are also encoded by the virus.
All encoded.
Here I am sitting here. I'm a big...
Is this fascinating to you?
It's no more than that. I'm seeing a ballet. Rich, I'm seeing like Polobolus. Do you know that group Polobolus?
Yeah. They wrap themselves around
It's a weird...
Yes.
It's they're great. And there are other groups like that too, but Polobolus, I think, is the best one of those kind. And if you were to give them the instructions for the adenovirus...
Oh, that's great. Maybe they can animate it for us.
12 replication.
That would be so cool to animate it with people, right?
Yeah. No kidding.
In a dance.
Well, they did...
I'd go see that.
We talked about this before. They did...
I would pay money to see that one. Martha Graham does adenovirus.
Cool. In the 70s, a group at Stanford animated protein synthesis.
That's right.
Alvin Ailey presents...
But this could be in a theater to music very well. Yep. Oh, that would be amazing.
And Joshua Bell playing in the back. Come on.
What would be the name of that dance?
Why can't we do this? The Dance of Life.
And we're not dancers. We're not even artists, Dickson.
This is a Greek tragedy because
if you told this idea to one of these dancers, they would look at you as if
no, I don't think so.
I think so.
No, because they do Debussy.
You have to find the right person.
They do Debussy music like this, and they portray spring or the fall or.
Go for it, Dickson. You got a new project.
After the Vertical Farm.
After the
you could have the premiere in the lobby of the Vertical Farm.
We could do that.
All right.
But
finally,
it would be wrong.
The last thing here is very important. There are proteins made very early as the DNA genome gets in the cell. They're called early proteins. If the cell happens to be sleeping, which we call quiescent,
that is not replicating.
These kick the cells into S phase because you need all the machinery going for the virus to take advantage of it. This is a theme we came upon last time. Viruses kick the cells into dividing.
Harry Ginsberg worked on a lot of that.
He did. The former chair who hired me here.
And me.
He hired you?
Yeah.
Well, he made
two good appointments.
That was you.
One of them didn't cost him a cent. That was me. That was just an adjunct.
What do we got next here? So, this is a summary of what I'm talking about, this kicking. The cells go through what we call a cycle, roughly every 24 hours, right? They go through a period of growth. Then the DNA replicates. That's called the S phase.
Right.
Then it prepares for cell division. That's G2.
Right. And
then mitosis occurs, which is what is diagrammed at the top here. Many of you may remember this from high school biology. All these different phases.
There are gorgeous pictures of all of this
where the
now
chromosomes line up
with
and
beautiful
segregate.
fluorescence and stuff.
And in the end, you have 2 cells. And that all happens in this little red part of the cycle. Very quick. And then the cells start to grow again.
And do their thing.
And if they happen to be resting in G1, there are some restriction points in G1.
And a lot of our cells in our body are just hanging out in G1.
Yeah.
That's
neurons.
They're waiting.
A lot of other cells.
Fibroblasts.
In fact, all of my cells are hanging out. And they're all restricted at this restriction point by a specific mechanism. And if the virus gets in and sees that it's here, it kicks them into S. And we're going to see, actually, one way that it does this. It's just fabulous.
So, could we take advantage of this mechanism, viral mechanism, to solve a medical problem without the entire virus infection being there? But do we have situations where host cells need to grow and they're not growing, and we can kick them into growth by using these early proteins?
How about the opposite? How about cells that
the
keep
opposite.
growing?
No, I like the opposite too, but what if you had a muscle damage injury and your cells wouldn't replicate enough to replace the damaged muscle?
I suppose you could do that. Maybe neurons that are damaged, you could kick them to divide.
Or repair, and grow between a damaged area.
Yeah, I don't know. It sounds good to me, but that's not my area.
I'm just speculating.
I like the other because when you have tumors, right? Tumors don't stop at these restriction points. That's the problem.
That's right.
They just keep going.
That's correct.
So figuring out these restrictions was really important.
Yep.
So adeno abrogates that restriction point. Okay, let's go on to our next virus. This is herpes viruses. I'm sorry, I should have shown the DNA. Lower left, bigger, double-stranded linear DNA genomes, but 120 to 220 kilobase pairs. Big, right?
Dude, you got the herpes virus bigger than the pox virus.
No, it's 130 to 375 is the Oh, you mean physically on the...
Yeah.
Oops.
Wow, these are
Well,
not
I
to
guess
scale.
they're not to scale.
Because look at the polyoma.
Right.
Yeah, right.
Not to scale.
Okay, not to scale.
And the herpes genome is full of origins. It's got 3 of them, as you can see here. They're called Ori-L, Ori-S, and Ori-S. 3 origins.
Question of just basic biology here. If forget about the attachment process as to whether or not these viruses are specific for cell types or not, because that's probably the case. But if you could inject them into any cell, will any of these double-stranded DNA viruses work in any cell, depending on, like,
another great question from Dickson. Holy cow. Great question. There are intracellular restrictions that can limit whether viruses will replicate in one cell or another. Case in point, polyoma viruses. If you take the mouse, compare the mouse polyoma virus and a human polyoma virus, SV40, they have sort of complementary host ranges. The mouse polyoma virus will replicate in mouse cells, but not in human cells. The human polyoma virus replicates in, actually, it's not human, it's monkey. SV40 will replicate in monkey cells, but not mouse cells or cells from other rodents. And in both cases, the virus can get into those cells.
Right.
And, in fact, can make early viral proteins. And so the infection gets rolling.
Okay.
And then something happens with the actual polymerase primase complex. So that for the mouse polyoma virus, the human polymerase or the monkey polymerase primase complex that's responsible for initiating DNA replication doesn't work right. Okay.
Interesting.
And the converse is also true. So right there at the point of initiation of DNA replication, there's an incompatibility in the 2 hosts.
Got it.
And interestingly, in the non-permissive host, what you get is an abortive infection. It even starts to change the cell, it transforms and that kind of stuff, and then the cell basically recovers and boots this sucker out, and everything's okay. Now, I'm telling you stuff that I knew from a long time ago. We're talking decades.
You know what? I must have heard this
that
one
story
time
is
too.
held up and probably been elaborated.
The basics are still similar. That's right. There are intracellular blocks. All right, let's check out these herpes viruses. Herpes simplex virus. There's the DNA at the bottom again.
Right.
2 identical Ori-S's and a unique Ori-R. The Ori-L. The Ori-L, it seems to be important for the transition from latent to productive infection. As you know, herpes viruses, when they infect you, they make it eventually to your neurons and become latent. That is, the genome is there, but it doesn't do anything. And it's thought that the activation of that ORI-L may be important for the transition. For example, stresses that activate herpes infections to become productive also seem to activate this origin. So that would make sense because you have to multiply to get more viruses. With these viruses, when they infect the cell, the viruses have linear DNA in them, but when they enter the cell, they're converted to a circle. This is one of the ideas for how these replicate. And then they replicate as a rolling circle. Can you imagine a rolling circle?
It gathers no moth.
Okay, so these go in the nucleus to do this, but they turn into a circle. Here's the model for that. This is actually derived from another bacteriophage. Rich, would you know which bacteriophage?
I'm guessing phi X. Is that right? Do I have that right?
Phi X and also lambda.
Lambda, right.
These are very, very commonly used now for genetic engineering.
That's right.
Or at least they were when I was
So on the
doing
left
it.
is your circle. So imagine the herpes DNA is circularized, and then a nick is introduced. Nick, right? And that serves as the starting point for synthesis. Now you have a little red DNA being made, and that's the polymerase laying down a new strand on copying the inner circle, the light blue circle. Then the dark blue, the other strand goes away, moves away somehow. It's probably melted off. And that's called continuous DNA synthesis. And then that strand that was displaced can then also be copied by another enzyme. And that's the discontinuous DNA synthesis.
You know, Vince, I thought of something else. There's a very popular show on television right now called How It's Made. You ever watch that show?
No, I don't.
Oh, you should watch that show sometime.
I'm not a big TV fan.
Well, you could learn something from it because they said tonight on How It's Made, we're going to show you how to make electric razors, storm doors, and guitars.
I'm sorry. I did watch it once.
Come on, they're great.
I was on the treadmill and it was on, yeah.
So, you could have a whole show about how it's made.
That is cool.
In the case of now they're viruses.
Well, we could do this for viruses, I agree.
You can get students to pay attention, is what I'm saying.
The only thing is, making a shoe, for example. I saw this one episode on how you make wrenches. It's amazing.
Yeah.
It's
they
pick
amazing.
Movies, but we don't have any movies of this stuff.
Yeah, but you can see them in your mind's eye. You said this is a rolling DNA synthesis problem.
Actually, you could get animators to work on it. It would be interesting.
Well, you already showed us a site for that last.
Yeah, ex vivos. Yeah. I think there's room for how it works
fantastic.
for viruses. In fact, today I bought the domain microbe.tv.
Should have bought sex.org. You would have made
No.
$1 billion.
I want to teach. I want to go, video someday with
Absolutely.
this stuff, and I want a microbe TV network.
I don't blame you.
Maybe it's internet based. Anyway, I diverge. So we
Not
have
really.
this. This is why it's rolling circle, because the displaced strand comes off and that gets duplicated.
Sure.
And this makes actually what we call concatemers, genome lengths linked together. Now, gentlemen, there is a mistake in this picture, which I picked up last night. This is from my textbook. There is an error in this.
We don't like that.
And it's pretty serious. If either one of you figure out what it is, I'll give you a copy of my next book.
An error?
I'm looking.
Should we play the dum
Shall. Are you smarter than a fifth grader?
Time's up.
Yeah, time's up. What's the mistake?
The fourth circle from the left.
1, 2, 3, 4. Yeah.
Discontinuous DNA synthesis. The arrows are in the wrong direction. They're going.
From
you're
5
right.
to 3?
On the template, it should be going 3 to 5. Yes.
You're right.
I can't tell you how many of these errors we pick up.
You know what? That's easy to fix too.
Why is that, Dickson?
You just on the visual, you just
but
reverse
in
them
all the books that have been printed, it's not going to be fixed. But when they sell out the printing, we can revise it.
That's why I only printed 1,000 of my parasitic disease books first, because a lot of friends wrote me back and said, I found an error on this one, and then I took all those errors into account.
Sure.
I redid it.
Yeah, we collect them. We send them to the publisher.
There's still errors.
They do 15,000 print runs at a time, and when they sell them out, which takes probably 2 years, they do another printing.
So, important point here. Concatemers.
Yeah, I'm sorry, we diverged.
Why? What problem do we solve here?
Yeah, that's right. What good is a concatamer exactly?
Right.
If I knew what it
You
meant,
got
I would tell you.
concatemers, and you cut it out precisely. The gap at the end problem goes away.
So these are cut.
Of course.
Each genome length, and you don't have this end problem. Exactly. Thank you,
And
Rich.
so when you redo this figure for me, Vincent, on the very 5' end there in the bottom right, put a little gap for me, okay?
Yeah.
So a question here is that nick always in the same place?
I'm thinking that's as a matter of fact, my herpes friends are, I think, in the process of debating whether during lytic replication there's actually a circular intermediate. I think that this is a controversial topic.
You're absolutely right. I didn't want to bring it in the book. We actually address this.
Right.
So there are some other models.
I mean, I think it's important to show this rolling circle model because it's important, and it may be true, but there's a lot of controversy about this at this point. So I don't know if they.
Not all of these stages have been visualized using.
I once asked my herpes DNA replication friend Mark Chalberg about this, and he said, referring to what the DNA must look like in the nucleus, he figured it was a tangled mess. Because you've not only got this going on, but you got a bunch of recombination and other stuff, and so it's probably a big network of interacting molecules. It probably really is a tangled mess.
This has been worked out well for lambda. Herpes is the issue. We don't know if this actually happens or not. I don't see why the nick couldn't be anywhere.
Right.
This model could accommodate that, but I don't know what the actual
So the next question is what causes the nick, of course.
Well, it's an endonuclease, right?
That doesn't answer the question.
I don't know if it's a viral or a cellular. But
Nice try, Vince.
Yeah, that's right.
You're talking to Dickson.
What do you want to know? Where the enzyme comes from?
Of course.
Cellular or viral?
That's still not the answer.
What do you want to know?
I want to know how repeatable this experiment of nature is as the virus enters a new cell. Does this happen in the same way each time, or does it is it random?
What is the nick?
The nick.
We don't know.
Don't know.
I don't know.
And
it
who
may
does
be
it?
known,
and
but
we don't know that either. So we don't know if it's random or not, and we don't know whether the host of the virus does it.
You have a list of the herpes virus proteins coming up, don't you, Vincent?
I do. Let's see if that's next here. Viral proteins for genome replication. The primase and the helicase, composed of 3 different proteins.
And I don't think that includes a nickase.
No. Processivity protein, which makes things go well. An origin binding protein, a single-stranded DNA binding protein, a DNA polymerase, 5 enzymes of nucleic acid metabolism, such as thymidine kinase.
Wow.
Okay? Not sufficient. So there may be the nickase from the cell. I mean,
is
cells
that a
have
real name, or are you guys just making that
No,
up?
I can call it a nickase.
Sort of a jargon.
Okay.
Nickase.
It's an endonuclease.
Endonuclease.
All right.
But it may be so. I don't know. It's a good question. I will ask someone, and you ask Dave Bloom. He might know, right?
You might know.
Or Chalberg, I guess.
Or
We
Chalberg.
have to get Chalberg on here sometime.
He would be terrific. He's the guy who figured out which proteins were required with a classic experiment of transfecting an origin-containing DNA into cells and then effectively transfecting in candidate genes until he trimmed it down to the 8 that would replicate the origin.
He did this slide. He did this slide, right? Yeah.
So what happens when you throw in colchicine and stop mitosis? And can you then infect those cells and get them to go to a certain point, but they won't go any further?
Colchicine stops the dividing because it screws up the microtubules, right?
That's correct.
So I would guess
my guess is it'll be okay.
It's probably okay.
Yeah?
But my guess, yeah. Me too, because the machinery isn't stopped. You don't have a checkpoint thing in there.
Got it.
Right?
So I don't know the answer to that question. My guess is they'd be okay.
Huh.
All right.
Yeah.
All right, this is Rich's baby, pox viruses. I'm gonna hand the baton to Rich.
I'm nervous. So pox viruses are weird because they most DNA-containing viruses go to the nucleus to replicate their DNA, where they can rip off all the nuclear enzymes, or as many of them as they want. Some need more than others. Pox viruses have decided to set up shop in the cytoplasm, and the way I describe this is that there are pros and cons to that. Pro is that you avoid the nuclear bureaucracy and you don't have to deal with all of that stuff. The con is that you have to encode all of the enzymes yourself, which I suppose if you go independent, that's not a bad thing to do. That's what pox viruses have done. So it's drawn on the bottom is the genome. It's depending on the virus, as short as 130 kilobases, as long as 375 kilobases. And it's a funny molecule because it's depicted here as double-stranded, and then these two things on the end. ITR means inverted terminal repetition. So the sequences on the left are also represented on the right in an inverted orientation. Plus, the ends of the linear molecule are actually covalently closed. It's DNA all the way around. So if you denature this thing, you get a circular single-stranded DNA molecule, but it's self-complementary. All right?
Got that?
I do. I got that.
Okay.
As you will see, this is all part of solving the famous gap problem.
Uh-huh.
So,
shall we go to the next slide?
Sure. Go to the next slide.
Ooh. Nice pictures, Rich.
Oh, yeah. I took these pictures myself.
Lovely. Oh, I like it.
Good, huh?
I do.
Not bad.
DNA factories.
Yeah, yeah, yeah.
Of course, I borrowed a lot. I borrowed the antibodies from my friends. Okay. So,
Dickson, there are no factories left in the U.S., but there are viral factories.
So we wanted to show this just, well, partly because they're pretty pictures, but also to give a conceptualization of this idea of setting up shop in the cytoplasm. So on the very left this is an infected cell. And on the very left, it's stained with a dye called DAPI that intercalates into double-stranded DNA and fluoresces blue.
It's gorgeous.
And so the big bright blue thing in the middle is the nucleus. Mm-hmm. And then on the left hand, there's two types of factories here. What me and my local friend here, as two types of factories, not necessarily generally in the field. At any rate, we need vocabulary to talk to each other. Around about 7 o'clock, you can see small, very well described circular spots.
Here, 1, 2,
I call
3.
those type 1 factories. I think that those are just getting going.
Uh-huh.
Okay? And then all around the nucleus elsewhere, there's a larger sort of amorphous, not so darkly staining thing. I call that a type 2 factory. So I think the type 1 factories are just starting DNA replication from an infecting virion, and the type 2 factories, this is an asynchronous infection. The type 2 factories have been going for a while. So then they're stained with an antibody to a DNA binding protein that's the product of a viral gene called I3. So everywhere that protein shows up in red in the middle panel. And then over on the right, the two are merged. So you can see that protein stains both the type 1 and the type 2 factories. And interestingly, the type 2 factories obviously are subcompartmentalized, right? There's little spots all over the place. So it's not like it's a homogeneous thing. There's a sub-organization in it.
What does that look like at the electron microscope level, Rich?
The factories at this point, they got all the cellular organelles cleared out.
Right.
So it's like a pretty uniform playing field. And early on, it's just a uniform electron density. And later on, you start to see virus structures show up in the factories. It's hard. One of the advantages of the confocal microscopy is it's very easy to see these subdomains. It's a little more difficult to distinguish one area of a factory from another by electron microscopy. Right. So the two techniques are really complementary.
Right.
So, and frankly, although the I3 protein is a single-stranded DNA binding protein, we don't really know what its role is in infection.
Is it essential?
Yes, I believe so. There's no TS mutants in it. I think they've done siRNA experiments to knock it down and show that it compromises the infection.
Okay.
All right, go ahead. You got another slide here? Yeah. Okay, so this is the same thing, same idea. This is just the merge. This one's stained with DAPI and viral DNA polymerase.
Right.
Okay, so this is the viral enzyme that actually does the DNA replication. One of the interesting things about this is you can see the type 1 factories are stained pretty heavily, and there's a type 2 factory down, once again, about 7 o'clock that's not stained much. Now,
down here, yeah.
I'm making this up, okay? No, this is not from the literature. I'm doing these experiments and thinking about this. I think that the DNA polymerase is staining where there's active DNA replication going on. And once that's done and you got a type 2 factory, the DNA polymerase goes away. But that's just my interpretation of what I'm seeing.
On the lower right, you have what looks like a type 2, very diffuse staining with polymerase.
Well, I'm not sure about that. These are confocal micrographs, so we could look at individual optical sections through that. I got a hunch that there's you're right. I don't know what that is. I'd have to have a closer look back at this and see if there's something piled on something else. Yeah. But you're right. It does look like at least there's a nucleus there, and it does look like maybe there's a type 2 factory with some polymerase in it.
Okay.
All right. So this is the model for poxviral DNA replication. And really, the model is based on just a couple of observations. One is that during vaccinia DNA replication, you can isolate concatemers. All right, so we know they exist. And the other observation is that at the junction of the concatemers, there is a structure that consists of base paired versions of these terminal hairpin structures. So I want to. It's important to emphasize that this is a model based on just a couple of observations, and nobody has otherwise seen this exact thing happen. So if you look at the upper left, where step 1 is happening, this is another picture of the DNA molecule, and you see these little bulbs on the end. Those represent these terminal hairpins, which are about 150 nucleotides long, and they are not completely self-complementary. They have extra helical bases, and they come in 2 flavors called either fast or slow, depending on how they migrate on gels, or there's other names. Oh, flip and flop. They're also. But the interesting thing, the interesting and important thing about them is that the 2 flavors are inverse complements of each other. So if you isolate them, denature them, and renature them, they form a perfect double-stranded DNA molecule, okay? And that's important for the replication. Also, the first parts of the double-stranded stuff, say between the bulb and into that little vertical arrow on the left-hand end and a little beyond, are inverted terminal repeats. So there are sequences that are repeated on the left and the right hand of the molecule, and that becomes important as well. This isn't a complete picture of the whole thing. It's but it's good enough for our purposes. So that the idea here, and this is the model, is that there is a nick introduced in the inverted terminal repetition on one end of the molecule. That's step 1. And if you do that and then take the hairpin and unwind it and extend it out, that's how you get that staggered thing. Right. Mm-hmm. And what's not shown there is the idea is that where the nick took place on the recessed end on the bottom of that molecule, that's a 3' end. Mm-hmm. So guess what?
Primer.
You just need a primer.
Yep.
Okay. So then you extend out of that with brown.
Right.
And since there are inverted terminal repetitions,
right,
they can pair with each other and fold back and make a panhandle structure, much the same as we saw with adenovirus. Okay? Now, that primer is now priming synthesis off of the original strand. And that can extend through step 4 and through step 5 and come all the way out to the end.
Got it.
And then ligate. And what we have is a dimer. And at the junction of that dimer over on the right-hand side where the little bulb is flip and flop base paired with each other. Okay? And then if you stretch that out and introduce a bunch of nicks and stuff, actually, since flip and flop, and since there's flip and flop and inverted terminal repetitions, you can actually form something that looks like a recombination junction, a holiday structure, and there's an enzyme that can resolve that from a concatemer to 2 monomers, and then all you got to do is fix the nick, and you're done. No gaps, dude.
Why? No gaps.
Primers.
Isn't that cool?
Okay.
Solves the gap problem.
It's a gap problem.
Tell you, they wouldn't sell many pairs of jeans here.
It works.
It
Been
does
around
work.
for a long time.
Absolutely.
So,
Absolutely.
but remember, the only two real observations are that there exist concatenamers and there exist these structures at the end. Everything else is, actually, people have observed nicks in the ends of molecules that I believe are even in the virion. So, and that seems to be a fairly, Dickson, for you, there seems to be a specific process. Nobody's followed up on that for a long time. But everything else is made up. Those are our models based on a few pieces of
Makes sense.
There
It
you
makes
go.
sense.
Somebody needs to work on it.
It makes sense. You can explore this by mutation, can't you?
what it really needs is a good in vitro system, I think.
Doesn't exist though, does it?
Well, there are pieces of it that have been reconstituted.
It's hard.
It's hard. It's very difficult.
All right. Beautiful. Oh,
Very nice.
here are all the enzymes involved.
Goodness.
So remember, this thing replicates in cytoplasm, so it has to encode all of its own functions for doing this. And here's a list of the enzymes that have been identified so far that are involved in this. And you can read the list. Polymerase, a processivity factor, the primase that lays down the... Oh, actually, we said there's no primers, but that's not... Actually, that's interesting. I hadn't thought of that. There is an enzyme that lays down RNA primers. Okay. So actually, in my mind, the implication of that is that the model that we just showed by itself may not be exactly true. And there is evidence for Okazaki fragments, little pieces of DNA. Okay. So even though you could do it that way, according to the model, there does exist a primase, implying that maybe the model is, there's more to it than that. Okay.
So how many open reading frames are there total for this virus versus how many proteins are accounted for?
There are 200 for Vaccinia, sort of the prototype. There are 200 open reading frames, and so we got a certain Well, there's probably half of those that are immune modulatory proteins.
Wow.
Where the virus messes around with the immune system to get around it. And another half that are, remember, it's got to do not only its own DNA replication, but it's got to do its own transcription too. It encodes its own RNA polymerase. It's a multifunctional enzyme, capping enzyme, poly-A polymerase, the whole 9 yards. Plus, it's this enormously complex structure.
Right.
The capsid itself is unlike any other virus. It's got about 70 proteins in it, but some of those are these transcription proteins.
It's almost an organism.
It's almost an organism. Doesn't have ribosomes. Okay.
Well, what's keeping us from...
What's next? Right.
That's right.
So at any rate, here's the list of the proteins that it encodes. And the
impressive.
little asterisk ones are the ones that we know are absolutely essential. If you knock them out, you don't get DNA replication.
So, Vince, what issue of Virology 101 is this?
What number is it?
Yeah, like 6 or 7 or
something.
8.
It's less than 10.
I know, less than 10. So in the very beginning, we began by nomenclature and by replication cycles and stuff like this. So now we could actually, if we wanted to, create a spectrum of complexity, beginning with the very simple viruses and working our way towards these things. And actually doing it as a bar graph or some other kind of spatial representation to show... You know, if you want to be a virus, you don't have to be very complicated.
Not at all.
You can be so simple, you can only make one protein.
Sure.
But if you want to be a virus that's almost a cell, this is what you're going to have to do.
Yep.
Sure.
So I think it's very instructive for us novices to have this spectrum of viral life, if you will, or viral non-life.
That's okay. Whatever you
the
want to say.
living dead, the zombie molecules, the zombie organisms, to line them all up and to see how beautiful and how rich this field is. Because, you know, when somebody tells you that I think I want to go into virology, you know, in the beginning I had that option. Why didn't I do that?
So, you know, we have these big posters in our labs of, you know, signaling pathways and metabolic charts for all these viruses.
I mean,
one for viral DNA replication, one for virus assembly. Sure.
I think that you guys have the richest imagination of all the biologists because you have to imagine how all of this happens because you can't see much of it. And you're doing inferential experiments in order to get to the data.
The viruses have the imagination. We're just trying.
It does. It does select for a certain type of investigator.
You know, I wanted to see my organism, so I picked a worm. That's the basic difference. But I still had to think just like the worm.
It's an interesting point. Yeah.
You guys think like the virus, and you say, okay, all of these are essential. Some of these are not. I can't believe that they're not all essential in some way or another, given a circumstance of.
Well, yeah. I mean, obviously they've been selected for. When we say essential or non-essential, we're talking about in cell culture.
Exactly.
But you throw these guys out in the jungle.
Oh, right.
And everything, all of these things have survival value.
So, Rich, what is your favorite pox virus that you work on? Monkeypox or?
No, the one I work on is vaccinia, the virus that
vaccinia,
was used as a vaccine for smallpox.
That has been around for a long time.
Yes, indeed. A couple 100 years.
Indeed, indeed.
All right, let's see what we have left here. There are a couple things to sum up, and then one interesting tidbit. Delayed synthesis of virion structural proteins prevents premature packaging. Okay, when you make a new viral DNA and you encapsulate it, it can't be used as a template. So viruses like to delay encapsulation, that is putting the DNA into packages for a while so they can make a lot of DNA, right? Makes sense.
It does.
So genome packaging is usually delayed, and this is controlled by transcriptional mechanisms that we'll get into later when we do transcription.
Packaging is spontaneous? Can you do this in vitro?
For some viruses, yeah.
You can take the capsids and the DNAs and just mix them together.
Yeah, sure, sometimes, but for the bigger, more complex ones. Every time you do that, it takes 15 minutes for me to edit it.
Well, then don't edit it. For God's sakes, just leave it in.
Okay, I'm going to leave it in.
This is part of life. You just have to cough every now and then. And when you have a show like this, it's hard to go for an hour without doing that.
So that was the point, delay. The second point, inhibition of cellular DNA synthesis. When DNA is carried out mostly by viral proteins, the cell is usually inhibited. The virus wants to increase the availability of substrates. It wants everything for itself. Right.
Phage T7 goes in and not only inhibits the host cell DNA synthesis, but it's got nucleases that chop the chromosome up into individual nucleotides, and it recycles all the nucleotides and uses it itself.
And it doesn't chop itself up.
Right.
And we'll talk about that another time. Adenoviruses, herpes, and pox viruses have this strategy to turn off the host. We don't know how, though. Sorry to say, we can't talk about how because we just don't know. Another generalization, DNAs are usually made in compartments. Rich talked about this for pox viruses.
Factories.
In the factory sense. Yes, here's an example for herpes. This is a cell, a single cell. On the left, either top is mock infected, the bottom three are herpes infected. This is stained for just DNA. And on the top, this is actually a nucleus of the cell, and you can see that's cellular DNA on the top. This is viral DNA on the bottom. It's very specific places. If you stain for a viral protein ICP8, you can see that it co-localizes with the viral DNA. That's very much similar to what Rich showed.
Very elegant.
Exactly. And finally, remember we said that the viruses want to kick the cells going, don't rest. We want your machinery active so we can make more. It's called exponential DNA replication. The virus wants to make as much DNA as it can. And this is sort of a tease for something we're going to talk about in another Virology 101. And there's a protein called the retinoblastoma, RB protein, encoded by the retinoblastoma gene, RB of the cell. And this protein is one of the controls over the entry into S from G1. It acts at this restriction point. Loss of this protein is associated with retinal tumors. That's why it's called the retinoblastoma gene. And so because of that, it's called a tumor suppressor gene. If it's present, it suppresses tumor formation. If it's absent, they occur. When it's present, it controls the cell cycle. If it's not present, the cells duplicate uncontrollably. They accumulate mutations as a consequence, and they become cancerous.
So the urge is to replicate.
Exactly.
And you have to put a stop to it.
Yeah, the default is to replicate, right?
That's right.
Now, viruses have revealed this mechanism of this protein. Let me just show you how that works on the next slide. This is one example. An early protein of adenovirus. Remember the E1A, which we said before, kicks the cells into S phase. How does it do it? It binds the RB protein.
This is very interesting.
It binds it. So here on the left, actually, not only E1A of adenovirus, but other proteins of SV40 large T, the human papillomavirus proteins, E7, they all bind RB. So RB is the gold protein here. I've lost my cursor. Normally, so here we have the purple is one of these viral proteins. RB is usually present in a complex with a transcription factor complex consisting of two proteins, E2F and DP. And that transcription complex usually binds DNA. And if RB is present, the transcription of genes that are needed for cellular synthesis and viral replication is inhibited. So the presence of RB is a brake on the manufacture of mRNAs encoded genes required for synthesis of new cellular DNA.
It's fascinating.
Now, when the viral protein shown in purple binds RB, it releases it from this transcription factor complex. And when that binds to the genome or the viral DNA, what do you think happens? You can make all these proteins that are needed for replication of both the cell and the virus.
Exactly.
So the virus wants RB off so it can have all these proteins so it can replicate its genome.
So, Vince, there are lots of other systems like this, too, in the cell for other functions. And one of them is called ID, which is an inhibitor of MyoD, which is a generator of muscle cells in fibroblasts. If you have a fibroblast, and you eliminate ID, it goes on to transform into a muscle cell from a fibroblast. It's quite amazing, actually.
Yeah. Well, the general principles are very similar.
They are.
Details that differ.
But to go back,
you can't go back.
You can't go back because you have about 4 or 5 genes that also program for muscle cell generation. So that's kind of built into the system because I think muscles are important. There are very few viruses that infect muscle. Rich, do you know any?
Thinking.
Coxsackie virus.
It's one of the most abundant cell types in your body.
Yeah.
And yet, it attracts very few parasites.
Coxsackie viruses will infect muscle.
They will.
There's some others as well.
Which muscle? Smooth or striated?
Both.
Both.
Yeah. Both.
That's why you feel so miserable with it.
Yeah, you have muscle aches. It can also infect your heart.
Cardiac also. So all 3 kinds. Or brain muscles in some of us.
Yeah, I have brain muscles, yeah.
I've got lots of that.
So in the cell, RB, you don't want RB always on this transcription complex. You need to go through and replicate sometimes. So there is a control, a normal control of RB where it's phosphorylated by cell cycle kinases, and it comes off the complex, and then you can make the proteins.
They should rename that protein now. That's a lousy name for
RB?
it. Yeah.
Why?
Retinoblastoma, because if it's a normal protein of cell replication.
I know, but it was discovered in tumors initially.
I know that, but
the absence of it was
it's
discovered.
not just important for...
Yeah, I understand. It's stuck, though, RB.
Understood.
And this is just one example. This was discovered, really, this regulation was discovered via these viral proteins.
Because it confuses the general audience, that's all I'm saying. This is a normal protein that regulates cell division. The absence of this protein.
Yeah, well, the name comes from its disease state. Yeah,
understood that.
Okay, I understand, but it ain't going to change, buddy.
Nope.
All right, I want to mention one more thing, just one more topic.
Okay.
So we've talked about making a lot of viral DNA.
Right.
But not all situations demand that. Sometimes there's limited replication. So if a virus is going to be quiescent in you and not do very much, you don't need to make a lot of genomes. So let's talk about that just a little bit.
You're anthropomorphizing, of course.
Yes, of course. I always do. Makes it quicker.
But it raises the question of
they don't want to do anything.
What's the difference between a rapidly and a...
Well, I'll try and tell you. Most DNA viruses like to undergo exponential replication.
They kill themselves, and they kill themselves, and they kill themselves.
Well, they kill the cells.
Yeah.
Where they make lots of genomes. But some establish long-term relationships in which the number of genomes is limited.
Is that like the herpes?
Herpes viruses, exactly. Human papillomaviruses.
Okay.
And there are various mechanisms. The DNA replication has to be regulated. So there is a mechanism for the parvoviruses, which we talked about in the first DNA synthesis TWiV. These are single-stranded DNA viruses, and the replication scheme is shown on the right. This virus needs one of its own proteins. It uses everything else from the host cell in order to replicate, but it makes one protein called Rep78/68. which is an endonuclease that's needed to do a lot of these nicks during the replication. It uses everything else from the host cell. Okay? Mm-hmm. These viruses will only replicate in cells co-infected with helper adenoviruses. They're defective. And the helper proteins of adenoviruses allow the parvovirus genomes to make large quantities of this Rep7868. But what if there isn't a helper virus around? What will happen? When no helper is present, you don't get a lot of this Rep7868, so you can't replicate the genome. So what happens? Little viral DNA synthesis, so the genome integrates into the host cell DNA and it allows it to be replicated there.
Sits and waits.
And if that cell is ever infected by an adenovirus, then it can rescue.
It rescues the virus.
But the genome survives. That's the key. Genome survives.
At all expenses.
Right?
No matter
what. So it integrates. So this is low-level DNA replication, not exponential.
What if at that point, though, it undergoes a mutation, which you can't rescue it after that? Does that now become one of these remnant viral sequences in your genome?
That would make sense.
Yeah,
that would make sense.
It would have to be in a germline.
Yeah.
This is mostly somatic cells, though. I don't believe there are germline parvoviruses. Not that we've detected. But you can never prove a negative, as Alan Dove says.
This is all true.
All right, one more example. This will be our last slide.
The papilloma.
Papillomas, both controlled and exponential replication from a single ori. The genome is a double-stranded circular molecule, and it has a single origin, very much like the polyoma virus DNA that we talked about last time. Mm-hmm. These viruses, of course, infect the skin, the epithelial layer.
They cause warts.
They cause warts and, of course, cancers, genital cancers.
Vaccinable.
Yes, this is the human papillomaviruses against which we have now 2 very good vaccines to prevent cancer.
Exactly.
And shown here in the picture is a layer of skin. We have the dead outer layer, and then we have a layer of differentiated cells. And as you go to the bottom, they become less differentiated. When at the very bottom, there are precursor skin cells. The virus is introduced probably by an abrasion or a cut into the very, very lowest level of cells, the basal
stratum germinative,
undifferentiated cells. They infect them,
yep.
And you have a very limited amplification of the viral DNA. It replicates as circles in the nucleus. So something keeps the origins down, and we don't know what it is. As the virus moves up through these cells, they become more differentiated. There's limited replication in those, but when it reaches the most outer layer of the most differentiated skin cells, then you have productive exponential replication. 10 times higher, 1,000 times higher levels of viral DNA than in the lower skin cells from a single origin. So it can detect, and this is controlled by the environment of the cell. These are undifferentiated at the bottom and very differentiated at the top.
Sure.
And also various viral proteins that interact with the origin. So it can go in a single viral genome. It can be either maintenance replication or exponential.
Did you have warts as a kid, Vince?
Sure did.
I did too.
My fingers.
Yeah.
I thought they would never go away.
They burned them off of mine.
And one day,
they
went away.
That's right.
I used to put all kinds of things on them, this acid to burn them off.
I think I've mentioned on a previous TWiV that there's yes, a matter of fact, we posted it at some point, an article written by Lewis Thomas.
Sure.
That elaborates on the observation that you can hypnotize warts away.
Come closer, Dickson. We can't see you. There he is.
You want to see me,
and
too?
That's Rich Condit.
Yes, it
is. Hi, fellas.
Hey, fellas. Let's do a few emails.
Sure.
And I've taken all the last names out because people can see this here. This first one is from Christina. Hey, TWiV guys. I'm currently attending Florida Gulf Coast University.
Where is that? Rich?
Fort Myers.
Fort Myers.
Yes, indeed.
Not a bad
I thought you were
place.
making a joke.
No. I just wanted to know which city it was in.
You know, that's the western coast.
It's above Naples, near Tampa?
No, I think it's south of Naples.
South? No.
It is south of Tampa.
It can't be south of Naples. There's nothing south of Tampa.
Is that right?
Naples, yeah. That's the Everglades below.
All right, so Christina's a biology major.
Yeah.
I need to get rid of her last name here.
We didn't see that.
I am taking molecular genetics in a virology course. I've learned that in a eukaryotic system, histones are bound to DNA through a force attraction between the negatively charged nucleotides and positively charged histones. The dissociation of histone proteins with DNA for either replication or transcription is due to ubiquitination, acetylation, or methylation, which decreases the positive charge on the basic amino acids in the histones. Knowing this, I wonder how viral capsid proteins dissociate from viral nucleotides upon entry. Is the mechanism similar to eukaryotes, or do the mechanisms differ from virus to virus? If the mechanisms differ from the eukaryotic system, what strategies do the viruses employ? Thank you. P.S. Looking forward to meeting you during your podcast on December 6th on our campus.
You're going there?
Rich and I are both going.
Excellent.
We are indeed.
We're going to do their last class. We're going to do a TWiV.
It sounds like they have a good virologist there already.
Yes. Sharon is the virology professor teaching this class, and she invited us to go down. And Rich and I are going to drive down in a yellow Mustang convertible.
There you go.
Well,
yeah, what's our dream?
Actually, that's our dream. We're going to have to put up with my Ford Explorer that's got 130,000 miles.
So I know a few examples, Christina, of how the genome is dissociated. For every virus family, it's different. For example, influenza viruses, as they enter the endosome and protons are pumped into the interior, that dissociates viral proteins from the genome so that they can get out. Semliki forest virus comes in also by endocytosis, and there the ribosomes seem to bind to the capsid and dissociate it, take it apart.
Gee.
Ribosomes dissociate it.
Cathepsins play a role here?
What's a cathepsin, Dickson?
It's an acid hydrolase.
I don't know.
It's an acid protein hydrolase.
That's a good question.
Proteins.
I would have to look. Nothing comes to mind.
Because when you encapsulate something into a phagosome or a virusome, I don't know what they're calling these things, and you acidify in the environment, it allows the fusion of lysosomes, which then releases all of these acid hydrolases, among which are cathepsins.
It could play a role. I don't know.
Okay.
For polio, the receptor takes apart the capsid.
Aha.
For adenoviruses, they also pass through endosomes. The low pH and enzymes in the endosome take apart the capsid. Reoviruses are taken apart in lysosomes by proteases. So they're all different. So it's not like a histone in a cell where it's one mechanism.
Right.
It's different. Every virus has evolved differently. Do you know what pox viruses do?
It's not totally worked out. There's an initial fusion either at the plasma membrane or in an endosome that gets rid of the outer envelope, but that leaves you with a core where the transcription goes on. And there's some old experiments that suggest that one of the early virus proteins is required for uncoating that the rest of the way to reveal the viral DNA in the cytoplasm.
Okay.
But those are old experiments. That needs to be looked into again.
So the answer to her question is yes.
Yeah, they differ.
Yes.
They're all different.
They're all different. Yeah.
And I'm glad she wrote, because now I've looked at Google Maps and I realize that Naples is, I've only lived here for 20 years, right? Naples is, in fact, south of Fort Myers, so we might have driven right past it in our yellow Mustang convertible.
If you go much further south than Naples, you're going to be in the Atlantic Ocean, I'm afraid.
All right, we're going to be surfing.
The Gulf of Florida, as it's called.
If we can get Skype hooked up and you're available, maybe you could join us.
December 6th, I'm going to be in Qatar.
Congratulations.
Thank you.
Welcome back, Cotter.
Yeah, we were actually, I've been there once, so this is a welcome back. So they're very interested in the vertical farm down there, Rich. So we're going to go down and talk to them about it.
Great.
Terrific.
Yeah, we're excited.
Well, maybe we could get Alan Dove to come in via Skype.
Sure.
That would be cool.
Skype,
Right?
that'd be cool.
That would be good.
All right, the next one's from Attila. I guess that's pronouncing it right. Dear TWiVers, as a Brazilian graduate student, I was very surprised to hear about the finding of vaccinia viruses in monkeys in Brazil in TWiV 95 and not finding out about it in Portuguese. So I wrote to the team that made their finding, interviewed them for my blog, and there's a link to it, and recommended their history to the local newspapers. Fortunately, they received divulgation in local newspapers afterwards. I guess the story was written in a local paper.
Sounds like it.
That's right.
I'm writing because I remember Professor Raccaniello asking how they came up with the idea of sampling monkeys at that point. So I asked them during our interview. They told me that already there were records of rural workers with vaccinia at the southeast region of Brazil, and some old records of detection dated from the 1960s at Belém, a city really far at the north region. So they knew that the virus was somehow spreading through the country, and they knew that similar viruses like cowpox and monkeypox can circulate in primates and rodents.
Right.
Also in Tocantins, the state where they collected the samples, a big dam was being built for the Lajeado hydroelectric station that appears at the episode picture, and a large region would be flooded, so the animals had to be rescued and transported to safe ground, a good occasion for sampling. Visiting the TWiV site to write this email, I saw the video of episode 96. I'm used to downloading the episodes directly to my iPod. It's very funny to have the opportunity to see the show being made and see the faces of you all. TWiV gets greater every time, and I really appreciate how you can have every kind of participants in this show, from graduate students and Professor Racaniello's son to Nobel laureates. It's fantastic to have contact with this sort of content here in Brazil. Keep up the good work.
Nice.
Right.
Nice.
It's a nice follow-up to that story.
Yep.
Yeah, so we'll put a link to this his blog, which in which he interviewed. It's in Spanish.
Yes, of course.
There you go.
It's a howler monkey.
Howler.
That's a howler.
Cool.
Have you heard a howler monkey? I won't imitate one, but they're very loud.
Dickson, I'm using Chrome, the browser called Chrome.
I know it.
And
yes.
when it reaches a foreign language page, it says, "This page is in Portuguese. Would you like to translate it?" So I'm going to click translate, and it's going to look.
Ow!
It translated it on the fly.
Hey, he's got a plaque essay here. Far out. I'm
cool.
using translate. I see. Yes, I'm
using Chrome
as well. Yeah.
There's a Tasmanian devil down below.
Oh yeah, yeah, right there.
There. Is that a Tasmanian devil? No, it's a pig. Sorry. It looks
Swine
like
influenza. Anyway,
don't have
thank
my
you, Attila. That's great, and I think it's cool that we can reach a lot of people who wouldn't normally have access to all these individuals.
beauty
That's
of
the
the
beauty
internet.
of TWiV, that we
That's true.
have contacts,
No,
and
you're
we
right.
can say, "Hey, guy, you want to be on TWiV?" And they come, and David Baltimore and my son and so forth. So
course.
it's cool.
Yep.
Dongoon writes, "Dear TWiV, I'm a big fan of TWiV since I bought my iPhone 4 last June. I haven't thought a podcast can affect me this much. The iPhone was a really good buy for me. That is only because of TWiV. I can make a call with it as well." Great. What a great ad for an iPhone.
They can make TWiV viral.
Anyway, I heard you introduced HTS on episode 95, high throughput sequencing. Or screening. Sorry, high throughput screening. I liked Alan's explanation about HTS. I appreciate it. The reason I'm writing this email is that I wish you can advertise a program funded by the NIH. That is the MLPCN, Molecular Libraries Probe Production Center Network. There's a link for it. I would say this program is a poor man's HTS, which the NIH provides all resources to screen a 300,000 compound library for your assay.
Wow.
You don't need to buy anything. Only thing you have to do is to develop an assay which potentially is compatible HTS and submit it. This is a free service, and it uses the RO3 and R21 mechanism. This is a grant mechanism. Right. This is open to anyone, even to foreign institutions. The centers in the network will screen for assay providers, and still assay providers receive money for doing follow-up experiments, etc. This is cool. The MLPCN has done more than 200 HT screenings for the public domain. The reason I bring this to your attention is the program supports HTS for infectious targets. Southern Research Institute is a specialized center in that aspect. The outcome of the screening is a probe which can be developed for a therapeutic candidate for antiviral or special compounds to see the effect of it in your assay. For example, if you submit an assay which can screen compounds inhibiting or activating a certain step, let's say inhibition of NP translocation to and from the nucleus, nucleoprotein. At the end of the screening, you will get a compound with that activity, and you may want to utilize the compound to study your basic science as well. The program accepts a variety of assay formats, such as a cell-based assay, fluorescence-based, or target-oriented or phenotypic assay as well. There is a chemistry center which works for the SAR and leads optimization for you. Let me get that right. Which work for SAR and lead optimization for you. What's SAR?
I don't know.
It's an acronym involved in the screening, which is escaping me. Again, everything is free once your assay is picked. I believe more virologists should take advantage of this program. It would be great if you can read this to your listeners or pick the MLPCN site for Pick of the Week so more people can see the program. I apologize for bad grammar. English is my second language, and please cut it back if you want to. I'd be more than happy to answer any questions. So Hoon is at the Center for Predictive Medicine and Biodefense in Emerging Infectious Diseases in Louisville.
Huh.
So there you go.
Nice. That's a very long, thoughtful letter.
I would like to use that. I would love to have a screen for inhibitors of, say, rhinoviruses, and then you can use them to probe the biology of the virus. That would be. I always wanted to do these and could never afford to do such high throughput screens. Okay, I think we shall cut it short there and move to a couple of picks of the week. And let's see what Rich has for a pick of the week.
Let me get down there. What the heck happened? Oh, there we are. Right. I'm going for Google Health.
Right? We haven't picked that before.
This is a new Google site.
Yep.
Where let me on this. Oh, I got it up here. Where you can basically put online for your own private use, or you can share it with others in your family or whatever, your medical records. Okay, so you can manage all your health information online. And it's got little features like setting your personal health goals or tracking your progress towards those goals. And you can, if you've got family members or whatever, you can share your health information. You can personalize the whole site. So, among other things, this means that anywhere you go at any time, you've got your medical records at your fingertips. And one of the things I like, is there's a lot of talk in the health industry about electronic medical records and that kind of stuff. And this is sort of Google saying, okay, let's quit talking and just do something, all right? See what happens.
Right.
And so they just put this online. You can use it. A doctor friend of mine pointed this out to me some time ago, and I said, I kind of looked at it, and it was sort of halfway interesting. And then I joined the Personal Genome Project.
Right.
And the Personal Genome Project, the way it's managing all of the health records is using Google Health. Okay.
Nice.
So I thought people might be interested.
Did you sign up for this? Have you been using it?
Well, I've got my record on. Yeah, I'm using it because it's. I was motivated to do it because, and I haven't done it completely. I was motivated to do it to link it with the Personal Genome Project. So my health information that the Personal Genome Project has is linked to them through Google Health.
Got it.
So you sign in and you get your page here, summary, wellness, problems, medications, allergies, test results, procedures, immunizations, insurance, and files. So you can add different things here as they happen. So you go to immunization, you add it, you can give a name, the date, and notes. That's cool.
Very.
All right, that's nice. I didn't know about this. Or maybe I didn't pay attention, but I might try it out. All right. Dickson, you got anything to pick?
Well, this is an old one. Not so old. It actually occurred last summer, but no one's picked it. So why don't we pick the entire Neanderthal genome to compare and contrast that with us? And it turns out that some of us got a lot of Neanderthal genes in us.
Where can we find the Neanderthal
That's
genome?
Actually, they published it in Science last summer. But I haven't heard much mention about it since it came out, except to say that we did hybridize, if you wanted to put it that way, with Neanderthals. Right.
Yeah,
so this might have implications for viral sequences.
Oh, yes, this is a good question. Dickson,
genes.
I'm sure people are at this moment looking for endogenous viruses and analyzing the data. I'm sure we will see papers on this.
Absolutely.
Yeah, that's a good pick. I like that because it's very interesting to know what endogenous viruses are there and how they relate to our current endogenous viruses in the human genome. Right?
Yeah, most anthropologists predicted that we were not related at all to Neanderthals.
Yeah. See, the anthropologists get a lot wrong.
They do, well, they were basing this on no evidence.
Yeah, I read a book whose name is escaping me where they really trashed the anthropologists for not understanding a lot of human beings. Oh, it was the Red Queen. That was a pick a while ago.
Right.
He just says they get human behavior very, very wrong.
I know. But there was one movie, I guess this goes along with it, called The Quest for Fire. Did you ever see that movie, Rich?
No.
Yeah, I think you picked that once.
I did once, but it goes along with this now because this was a French movie in which they had to invent two completely different languages from scratch. Neanderthal-ese and Humanese.
And they made it in the movie, right?
And they made it in the movie, and they did. They actually did. The Neanderthals actually mated with humans. And they didn't show whether they produced viable offspring or not, but they didn't raise the big issue of what led to the demise of the Neanderthals. They didn't raise that issue, but I think they got killed off by our microbes that we brought up from Africa during our migration out of Africa. So we exposed them to a whole bunch of things because the genome of Neanderthals shows a very restricted MHC complex. It's very narrow.
Restricted population,
extremely narrow. And so it looks like the same situation that happened when the Spanish came to the New World with all their microbes and brought back syphilis, of course, to the Europeans.
Where were the Neanderthals principally
Well, they were scattered throughout Europe.
Oh, yeah, but not Africa.
And into Asia. Never into Asia, Africa. Never.
But a bit in Asia.
A little bit into Turkey and places like that, but that's where they stopped. Okay?
And when we moved from Africa to Europe...
As the Ice Age retreated, allowing us the freedom of moving out of Africa, we invaded Europe, of course. Where else? And even there's no evidence for armed conflict between Neanderthals and humans. So we didn't kill them off by, strangling them or poisoning them with darts and stuff like this. We killed them with kindness. We mated with them, for God's sake. How, some of us must have been fairly desperate. I can imagine.
Hey, we don't know what we look like back then.
That's true to us, I'm sure.
But beauty is in the eye of the beholder, Dickson.
Isn't it? And in a dark cave, we won't
Dickson.
go there.
All right, that's enough of Neanderthals.
But anyway, I'm fascinated by the concept of Neanderthal.
All right, well, my pick is one that was suggested by Eric, who wrote, I found a YouTube channel, and the nerd in me almost had a heart attack.
That's a great way to phrase it.
Because it's so cool. It's basically a whole database of lab techniques like Western blot, real-time PCR, Bradford assay, etc., from Abnova, the world's largest antibody and protein original manufacturer.
Cool.
This would make a good pick of the week, since I noticed on your virology blog that you said a lot of people have been asking about the things done in the lab, which is why you put up the toolbox.
Here.
Unfortunately, there aren't a lot of virology-specific videos, so he gives us the link, and he says, I'm going to have fun watching these today. So this is a real science nerd, right?
You can have fun watching a Western blot evolve.
So this is a good channel. It's all these
I like it.
Techniques, electrophoresis, clonogenetic assays, his-tagged protein purification. So there you go, YouTube videos, a whole bunch of them.
Fantastic.
No plaque assays.
No, Rich, you and I should do that.
We're gonna have to put up a plaque assay.
Put one up.
Yeah, we should do that. I could film you, and you could film me.
No, reverse transcription.
See that?
Look at this. Immunofluorescence. Hey, I ought to look at some of these.
Come on, it's
so
good
they're cool.
stuff.
So thanks for that, Eric, and that's our pick of the week.
Okay.
Excellent.
And that will do it for another TWiV Virology 101. You can find TWiV on iTunes, the Zune Marketplace. And if you're new to TWiV and you go to those places, leave a comment, especially on iTunes. Helps us stay on the front page of the medicine podcasts. Yep. If you have a smartphone, you could listen, and specifically an iPhone, you can listen with the Microbe World app, which streams the episodes to your iPhone or iPad. You can find it at microbeworld.org. You can always go to twiv.tv, where you can play the episodes or download them directly to your computer or iPod.
Good.
And, of course, we love getting your questions. Send them to twiv@twiv.tv.
Yep.
And our next episode will be all listener questions.
Outstanding.
All right.
Because that will be 107.
That's a good deal, Vince.
Because we have so many of them, we need to get through them. But we love to do it.
I love it.
Dickson Despommier, thanks for joining us.
Thanks, Vince. And Rich?
Dickson is at verticalfarm.org.
Yep.
And soon to go to Qatar.
Correct.
QATAR.
That's it.
Well, I hope you build a vertical farm there.
So do we.
Rich Condit is at the University of Florida, home of the slumping Gators.
Now. We just beat Georgia.
You did? I watched the game, Rich. I thought about you the whole time. It was great.
Yes, we did. We beat them in overtime by a field goal. And we're coming back.
I turned off the Notre Dame game the moment I knew it was going to end in disaster.
We're going to be okay. Everything's going to be okay.
You're going to be fine. You're going to be just fine.
Well, you're in Paris, Vincent. I'm going to be in the mountains of Virginia, up above Blacksburg.
Have a good one.
What are you doing there?
It is a weird transcription meeting.
How about that?
Fancy Cap? Are you going to Fancy Cap?
Actually, it's regularly attended by Max Gottesman.
Nice.
Oh, yeah.
This is post-transcription, post-initiation events and transcription.
Great.
It's all of the rejects from mainstream transcription
That'll
meetings.
Be a lot of fun. Well, thanks.
It's a great meeting.
Thanks for joining us, Rich.
Yeah, great to be here. Good fun.
We'll see you in about 10 days. I'm Vince Racaniello. I'm at virology.ws. And this will come out as a... Do you want to leave, Dickson?
I do, actually.
All right, we're almost done. Just give it another minute.
You better hurry.
I'm sorry. You want some water?
No! That was the cruelest of all cuts. You guys are so bad. I'm not watching my watch. I'm watching
This will come out as video, hopefully, so you can have the audio or video, and you can watch Dickson turning red. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.